Welcome to this free webinar on bioinformatics for next generation sequencing. This program is an introduction to the field of bioinformatics, which is an intersection of informatics and biology. In this program, we will discuss the changing landscape of data-driven research in life sciences and the opportunities that come with the widely accessible high throughput technologies and computational methods for analysis and annotation of such detailed data in research and industry. We will discuss various emerging technologies and their impact on healthcare, agriculture, environmental sciences, public health, and more. Omicslogic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today and throughout this program is result of an hard work by an amazing team led by company co-founders Dr. Alfred Tauber and Dr. Leonid Brodsky. So Dr. Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation. And he is recognized internationally for his generous support of many research projects and initiatives. Dr. Brodsky is the director of the Tauber Bioinformatics Research Center at the University of Haifa, and he's an expert bioinformatician and biostatistician. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team, as you see here, includes experts in bioinformatics, computer science, and training. And some of our team members would also be presenting today, and I'm going to be introducing you to them shortly. In the upcoming Omicslogic program, you will have the opportunity to interact and collaborate with an international team of experts. And I want to introduce you to our team and the mentors who are going to be mentoring this Omicslogic programs. We have first Ilya Brodsky, who is our co-founder and CEO at Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Julia Panov is an Omicslogic mentor and an expert bioinformatician who has developed many of the resources that we use for the Omicslogic programs. Dr. Harpreet Kaur is also our Omicslogic trainer who will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning, and we will get to meet her soon. We also have Dr. Mohit Mazumdar, who is a Omicslogic project mentor and an expert in big data and computational biology. And I know that many of you have already met Dr. Mohit. And now I want to invite Dr. Mohit to take this session forward. Dr. Mohit, over to you.
Thank you, Biksha, and hello, everyone. All right, so how many of people? Okay, so we have a lot of people, and hello, Dr. Prabhuddha. Nice to see you here. So before we talk about the session itself, I would like to introduce myself. So a very good morning to all of you. My name is Mohit Mazumdar. I'm one of the global representatives of Pine Biotech. And in my current role, I'm collaborating and working with universities, academia, research institutions, and industry to provide bioinformatics education and research support. I have 12 years of experience working on different bioinformatics projects during my PhD, and after that, I joined a company which worked directly with pharma industry. So I have been collaborating with pharma healthcare industry to solve high priority scientific questions related to biomedical data analysis and also bioinformatics capacity building. So that's about me. And I see, as I was telling, that a lot of you are joining and have joined. So I would love to know about you as well. So if it is possible for you to type in the chat, like Mead said, hi, and would you be able to write about something about yourself, like the degree that you're doing or the specialization, and like if you're doing masters, if you're doing if you're undergraduate course, if you're doing a PhD, if you're in postdoc, or if you are in just finished high school, and or if you're in high school. So can you write that down for me? Thank you, Bishwajit. Thank you for sharing. So you're doing BTech biotechnology, and in which year you are? First year. Excellent. Okay. Achintya Chakraborty, first year, BTech biotechnology. Wonderful. MSc bioinformatics, second year. Prithviraj Paul. That's excellent. So we have a bioinformatician in here. Really good to see that. And a lot of biotechnologists. That's again amazing. Prashant. Hi, Prashant. Postgraduate in biotechnology. Excellent. Shushmita is again MSc biotechnology. It's great to see so many biotechnologists amongst us. And what we are going to discuss today will be a lot more impactful. Now that I know there are so many biotechnologists, it makes very much sense for you to be able to go through this session and do some of the exercises that we are going to do today. Hi, Nupur. Good morning. So you are MS Pharma. That's amazing. So pharma. So we are going to talk about also pharma industry and what it is right now, the landscape, because I've been collaborating, working with a lot of pharma companies. I think it will be useful for you as well. Apala, hi Apala. So you are BTech biotechnology, fourth year. That's good. Soyani Mazumdar is BTech biotechnology first. Arpan is BTech biotechnology final year. Thank you guys for this introduction, and I'm telling us about the specialization. So before talking about the topic itself, let's discuss why we want to learn about big data. What we are talking about big data, bioinformatics. We're talking about next generation sequencing, and this exciting technologies, and we are hearing about machine learning, data science, all those things. So let's just like break them down one by one and see how like it sounds really trivial and complex, because there are so many things happening. But how can we get to learn in a way that we understand this holistically and understand this also in depth? That's very important, right? So today's session will include an introduction to Omicslogic resources, and that will be followed by hands-on practice. So again, I'm going to ask you another question. So before starting this session, it would be really nice to know that how many of you are on laptop, desktop, or cell phone or others. So because I think the hands-on session would require you to something like good to have, like you can write 1, 2, or 3, 4. Just not, you don't have to write the entire laptop. So just desktop, laptop. That's good. That's good. That's good. Just waiting for cell phone. Okay, good. Okay, that's not a problem. So we will be recording this session as this is getting recorded, and we'll be sending you. So if you're not able to do the hands-on, then if you have a laptop, then you can join via that also, and you can have like multiple things on cell phone and laptop as well. But obviously it's much easier, I guess, to do it over the cell phone to join a meeting. But to do hands-on, we recommend that you use a laptop or a desktop. but no problem. We can do that later as well. Okay, lot of people have shared. Thank you, thank you, Srijita. So you're on cell phone. Okay, so about the session and big data in biotechnology, and bioinformatics and biology. So these fields, like life sciences fields that we're talking about, may that be microbiology, may that be agri-biotechnology. So all of this domains are generating data, and when we talk about generating data, it sounds like, it sounds very vague. So all the experiments that you do, all the experiments that you do in the lab, the outcome of that experiment is some sort of results that you get. That's data, right? So when we were doing single experiments, like we are doing a cloning, we are doing expression, then we are doing purification. So we are working on single gene. Maybe we are looking for a protein, and we want to see that if we get that protein purified or not for further study. But it's a single experiment that you're doing on a single protein. So that's very important. Now, what has changed and what has happened during this entire, a decade or so? High throughput technologies. So genomics revolution. So that all happened after the Human Genome Project. So we got to know about the human genome, the entire genome, 3.2 billion base pairs, quite a lot, and number of genes, number of proteins. And then we started looking into other organisms as well, to learn about the insights. And now, because it has become like lot cheaper, lot cheaper that than what it has started from. And one of the reasons for it to become cheaper is its application, huge application. We are going to talk a little bit about that also. So biology as data science. Okay. So why biology as data science? Well, the reason is simple. Biology is rapidly acquiring the characteristics of data science. Billions of data points on genes, proteins, and molecules, because I'm talking about high throughput. So 3.2 billion base pairs, 20,000 genes. And I mean, if we talk about single cell, then you can imagine. So billion of data points on genes, proteins, and other molecules. They are now compiled in large files, and they are systematically studied. This influx of data, and associated methods and data-driven research makes bioinformatics for life science as prominent as the emergence of computer for the tech industry. So I'm doing a really big comparison here. I'm trying to tell you this because computers have evolved the tech industry. Like, it has changed the tech industry entirely. And when I'm talking about bioinformatics for life science, then what I'm trying to tell you is that the data that have been generated and that's getting generated is as important as that. So you can imagine. So analyzing this data will lead to more knowledge and understanding about us. Understanding about living organisms. And we have this introductory session that we are doing today for an overview of bioinformatics for students, faculty, biologists, and clinicians. So why? Again, the question is what and why. So big omics data is now shaping modern biomedical innovation. So we are talking about pharma, we are talking about healthcare industry, we're talking about diagnostics. So it's shaping the entire industry because we are using precision diagnostics, we are using big data integration, and then we are using machine learning to transform personalization of treatment selection. And then there are innovative algorithms that are coming up to allow this unprecedented capabilities for drug discovery, design of large molecules, drug repurposing. Someone wrote about cheminformatics. Yes, huge application in cheminformatics. So these are some of the areas that there is a direct application of bioinformatics. So mechanistic studies, we do. So I did my PhD from JNU, and over the time of my PhD, I collaborated with 40 research groups, 40 different research groups who are working on either mechanistic study, or they were doing some either they are looking at drug response, whether they're looking at drug target, whether they're looking at a mechanism of a protein, how the protein works, how it interacts and stuff like that. So application is there, bioinformatics application is there, all this area. In clinical scenarios, the applications are huge nowadays because of the things that I discussed right now, which is like personalization of treatment selection, how that works. Biomarker discovery, when we are talking about drug discovery, when we are talking about this outbreaks, when we're talking about viruses and vaccines, how we can use bioinformatics, and bioinformatics has been applied to all of those areas. And who are doing it? I mean, these are some of the biggest company on earth, on pharmaceutical and healthcare companies. These are all pharmaceutical and healthcare companies, and they now have, all of them have huge groups of researchers, scientists, and clinicians that are working together to solve this data-driven challenges. They're generating data, they're solving data, they're looking for patterns in the data, and then with that pattern, they're coming up with solutions. So one of the example is must have heard about this company because of it's US based, and I think Donald Trump talked about it a lot also. So Regeneron, if you can see, 1 million exome sequence, 1 million is the benchmark they're starting with, and this is just the beginning. Like they're saying that this is just the beginning, and they're just at the tip of the iceberg. So the impact of these sequencing in human health and novel drug discovery. So this is just talking about the exome, the exons, the coding region. There are several studies, there are several domains with which we are integrating different omics domains. So it's just getting more and more exciting, and it's important to learn about this exciting stuff, because this is somehow be the future, I guess. At this point, there is no way that you can ignore the importance and adapt to this newer way. So it's good to have this knowledge and good to have this understanding. And to do that, I mean, so it's clear that the rapid growth of high throughput data has given rise to this data-driven discovery in life sciences. So as a result, there are a significant demand for data science skills and experience with bioinformatics methods of analysis. But what are the challenges? Because it's in demand, but I mean, what are the challenges that are there to be able to learn this, know this? Because one of the biggest challenge I see is that the interdisciplinary nature of this domain. So physics is related, biophysics, biochemistry, biology itself is like a detailed knowledge of everything that is going on. So that's a challenge in itself, that to be able to learn all of that. But you don't have to learn actually all of that. So what you have to, I mean, in my experience, what one has to start with is to look at a specific problem and understand it in depth. That's what research is defined. That's how I can just define research. So let me ask you in general, how do you feel about this entire thing, and what is a challenge for you currently? And if you want to express, you can write it down in the chat, that what is for you is a challenge. But to talk about the introduction to this kind of domain, fields, and interdisciplinary work, every day we are working with students, and we are working with researchers, and we are working with faculty from around the globe. And what we are doing, we are trying to simplify bioinformatics and overcoming the difficulties with innovative curriculums, cloud computational infrastructure, to overcome the burden of having a really expensive computational infrastructure to be able to do this analysis. Content development. So we are doing publishing supplementary study materials, and we are publishing courses and research projects which are based on hands-on bioinformatics experience. These are some of the ways we are trying to contribute and help uh, transform our knowledge and apply that into a translational sciences, and what is actually is helping the society or the mankind or humankind. So it's a small contribution, but we are trying to do that, and we are, these are some of the resources that I wanted to discuss and review with all of you, whosoever are new. Many of you have already gone through these resources. So these resources are built to learn, like these are courses, online courses, of course, and that you can learn at your own pace. So there is no timeline as such. So the course provides introductory materials that have been taken by thousands of people around the world and rated really high. And we have got like really a lot of feedback. So these online courses like Introduction to Bioinformatics is a free course that I think you can take, and whatever I talked about today, like at the beginning, talked about data-driven discovery, we talked about personalized medicine in diagnostics, in treatment development. It's a free course, so just you have to just browse through it and you can learn, and we'll share the link in a while. So as I was telling you about the courses, that it provides perspectives from experts on this exciting field. Each course has like a quiz that allows you to see how much you have learned. It's just to test your own knowledge. And the platform itself, the platform that we are going to talk about, the first platform, we are going to talk about 3 platforms, and we're going to do some hands-on together in this session. But in the first platform that we are going to be using in a while, we measure all the activity using a point system. Here you can see there's a point to identify this entire thing helps us to identify technical issues and resolve roadblocks that are common for bioinformatics problem, and people might have, what problems that students can get, what problems faculty can have, something like that. So it's a system to help this entire process. There are quizzes, as I was telling you. This tells you exactly how much you have done and what you have learned out of that exercise. And in the end, a certificate from the program mentor and Tauber Research Center, University of Haifa, Israel, Pine Biotech, US, and the Collaborative University. If you're in collaboration, if you're in a collaborative program, then that's the certification. That kind of certification is what you obtained after finishing a program. So certificate of completion, excellence, peer-reviewed project, all those things. So talking about projects, if you ask what is project-based learning or project-based training, what we do is it's an experience of learning real application of bioinformatics. Why I said so? Because why this is a real experience? Because we are taking data sets that are relevant from high impact publication, and they are well digested in the society or in the scientific community, means that they have an impact on what they have done with that data set, and convert them into practical exercises, and then develop them into research projects. So we take publication data from there, methods, we define methods using our platform, and then convert them into tutorials that you can take. So what you're doing is that you're going through a publication that did let me not say a publication, you're going through a research that a group in Harvard or group in MIT, or like all the good schools, they did some good research and they published it, and we are now we convert this entire publication into a tutorial that you can go through and actually do what they did using a very intuitive and easy T-Bioinfo platform. So that is the second platform that we will be talking about, and you will be also doing some hands-on practice today if you stay with us. Okay. So the focus and specialization, we want, as I was telling you about the translational aspect of this. So the translational aspect is that application of this technologies into oncology, for example, if you're doing precision oncology, virology, infectious diseases, you can work on neuroscience. You can work on neuroscience and neurodegenerative diseases and whatever your field of interest is. Agriculture, that you can plant biology and you're generating data from like sequencing of different plants and stuff. So that's there for specialization because it's a multidisciplinary field and it combines, so the data is being generated in all of them. So the purpose is to get to a place where we will be able to do independent projects. So this is a broad overview that gives, it's a journey from developing the concepts to developing your own project. And using the same analogy, because you get to train on different data sets, and then you get like an idea, because that's what I have also figured out, because if you go through examples, it's easier to start implementing and developing. Then you have ideas, then you get ideas, because you know that how this entire thing has been done, rather than just starting from the very beginning without any knowledge. And that's what most of us have done in our PhDs. And as I was telling you, that a lot of reviews and a lot of people have said good things about this entire training. And the first platform today, as I said earlier, this session will include practical. So the first platform we are going to explore is edu.t-bio platform. In order to access some of the free resources that we will be providing you, and you will have to create a login account, free login account. So let's go. So let me just pass it on to Deepsha. She will take you to the platform and help you get registered on the educational site. So over to you, Deepsha.
Thank you so much, Dr. Mohit. I will share my screen, and I hope that you can share the screen and see the screen and do this with me. So how many of you are on the link that is shared on the chat? If you are there, just put 1 in the chat box so that we can understand that you are here on edu.t-bio.info. Okay, thank you so much for letting me know. So if you're joining us for the first time and you have not had an account, if you already have an account, you can log in using the details. And if you're just starting out, just go to register. Once you go to register, you can register with your username, email. A password won't be generated automatically. Once you put in a username, email, you check this box and you enter your first name and last name and click on sign up, you will receive an email from where you can reset your link, or you can also connect with all of these other social login details. So please go ahead and do this. So we will take just a few more minutes so that you can get started. So if you are not registered, click on register option again here, and you have several different options to log in. If you already have an account, then please log into your Edu.T Bioinfo account. So how many of you are there? Because last time we said it was number 1. So now we will put number 2 on the chat if you can complete the registration. So let us do it together. Okay, Bishojit, thank you. Okay, wonderful, Mohua. At least we need a few more people so that we can progress together. You can also do this in case you're not able to do it right now. You can also do it after the session and watch the video. This is a free registration, and once you're registered on this platform, you do not have to keep on registering again and again. It will be the same user ID and login that you provide, unless you're changing your email ID. Okay. Anyone else? Okay, so once you are registered into the platform, you will come across different resources. Okay, Nupur, if you're having any problem, we can solve that. It could be anything, Prithviraj. Your username could be anything that you would want to give, but make sure that it is something that you can understand. Later on, you can also change your username to be your full name. So we will be talking about profiles a bit later. For now, what I wanted to show everyone is this. Thank you, Achinta and Shushmita and Shubhadeep as well. So, okay, so a few people are there on the platform. So we will be looking at some of the resources that you get access to once you join, which are free resources. So anybody by creating a free account can access these resources right away. So the first one is the course that Dr. Mohit has been talking about, which is Introduction to Bioinformatics. So let us all click on Introduction to Bioinformatics. Okay, once you click on Introduction to Bioinformatics, you would come across this course. So once you come across this course, you will be seeing an overview. Nupur, we will be helping you out as this is a session which we're doing with everyone. We will be reaching out to you post the session, and you can also email us post the session, and we will be helping you out from there. We just want to move forward with the session with everyone else, and you can rewatch the video and do it later. So here you will find the course description and also a video on what the course is about. So once you go into the course, so it shows me that you don't have to retake the course because I've already done it. But once you go to the intro survey, and I know that I'm not logged in, so I'm not going to be showing everything that is there. So in the intro survey, the first thing that you do is fill up a survey. So this survey helps us to know more about you, your interest in research, and what you're interested in learning about, as we know that people are coming from different backgrounds, different educational and research interests. So that is where it is helpful. And then you have different sections of this, which will help you learn more about the different bioinformatics approaches. You see the subheadings like bioinformatics in healthcare, what is translational bioinformatics, something that Dr. Mohit was talking about earlier. So please go ahead and take this course at your convenience. So this course is always going to be free, so you do not have to take it right away, but we wanted to introduce you to this course. And also we have a blog where we keep posting about many informational topics. So if you go to our blog page as well, you can find a lot of resources on different topics. We have a lot of resources, lots of things about our training platform. We also have launched a platform which you can learn R and Python. So we will be talking, and Dr. Mohit and Dr. Harpreet will be walking you through the rest of the resources very soon. So keep exploring these resources for now. And I would want to pass it back to Dr. Mohit. Dr. Mohit, over to you.
Thank you, Viksha. And I hope everyone is able to log in and create that account. So talking about some of the resources and what are these resources and what are you going to do about it? I mean, you see the resources, but what next? So in educational platform, you will find interactive content to learn various omics technologies. Okay. So if you're interested in learning about DNA and the changes that can happen to the genetic code, the genomic series of courses explain how next generation sequencing can be used to study genome-wide genetic variation that results in phenotypic changes. So these are the three courses that we have for genomics right now. And if you're interested in learning about RNA and the expression of proteins and stuff, so RNA-seq analysis. So this is very popular because this has a huge application in pharmacology, pharma industry, and in biotechnology. So This is a complete guide to RNA-seq analysis. Transcriptomics 1 course is an introduction to next generation sequencing to study the RNA expression. Transcriptomics 2 is a course about gene expression analysis, and it focuses on studying variations in expression between conditions. So it uses a lot of statistics, it's a lot of statistical principles. These are all starting from basics. So if you don't know anything, you can still start from the very basic and learn from the basic to an advanced level. So the first one takes you to about to the gene expression understanding, and then their statistics, their t-test, their factor regression and analysis. And transcriptomics 3 is the most exciting for me because it has the application of machine learning, but it requires you to learn 1 and 2, and then you have a deeper understanding of this entire. Transcriptomics 4 is about single cell. So single cell RNA-seq, many of you might not have heard about, but it's a very powerful technique for classification of tissue-specific cells and the study of time course data for thousands of single cell samples. So it's a it's getting like it's a very modern technique, and getting really popular amongst scientists to discover new things. Metagenomics, and many of you might are coming from a background of microbiology or are interested in virology. So I think metagenomics is an excellent place. Okay, Prithviraj, I think, yeah, Bicha, thank you. So as I was telling you, so you can dive into the world of microbes and learn about microbiome in this 3-course series. So introduction to metagenomics will familiarize you with microbiomes influencing their host and composition of unique communities, these bacterial or viral communities using next generation sequencing. So one of the advantages of learning about or doing genomics is the way we can study samples like directly. We don't have to do all those steps that we used to take, and that was one of the roadblocks. Now we can study samples right from the soil samples, gut samples, all kind of studies are related. So it has a huge influence in human health, and you will learn about 16S RNA DNA sequencing, microbiome composition in different conditions and stuff like that. And then data science and machine learning. So many methods in bioinformatics requires in-depth understanding of various machine learning algorithms and tools. So in this specialization tracks, we develop a series of courses. So it gives a solid foundation, but it does have a component of statistics and mathematics. But I think it's easier to understand because we have created this entire platform thinking about biologists, biotechnologists, and clinicians, those who have not a background of computer science and maybe mathematics. So it's built for everyone in a way to learn. And after you're done, like after you have learned through these courses, the best thing is to apply the understanding and the knowledge. So these are the projects. By seeing, you will be actually, it's an experience because by seeing how experts have analyzed their data and achieved research results, you can start developing a research question of your own. That is what I was talking to you about before, and identify the appropriate type of data and planning your analysis. Because look, I mean, I'm seeing there's a lot of data, there's a lot of research you can do, but there is a limitation also. You cannot do every kind of research using that data. So you have to learn that this kind of data will give you what kind of insight. You have to gain that through experience, through working through these projects, and maybe your own interest, you're learning from publications and stuff. And it's important, many of you are in first year, and I have heard people saying that it's too advanced for us because we are just learning about central dogma. We don't even know some central dogma. We don't know many of this. What is a biomarker? These are like really stuff that we have never heard about. But what I've realized is that it's not about learning and knowing everything, because it's impossible actually. Because it's, even after so many years, I'm like amused all the time that what all is happening. So it's okay to be like that. It's okay to be overwhelmed with this all information at, even at an early stage. But it's important to get an idea, or it's important to get into that mindset of what research is. I mean, how do we find something new? Because you all will be learning the same thing that we have been learning for years. But when it comes to jobs, when it comes to I mean, some translational aspect, like you have to apply this knowledge to get something. It's required, you that's where you're required to know stuff. So learning at the beginning, at the early stage, if you're in first year, that's the best time to start this, because it takes time, right? That's what I meant. So the introductory courses leads to multiple courses and projects that serves as a springboard to this world of data science, bioinformatics, machine learning, and whatever is happening in healthcare and biotechnology and stuff like that. So the second platform, our second innovative platform, is developed at the Tauber Bioinformatics Research Center, University of Haifa, as Divsha was telling you. Dr. Leonard Brodsky and his team, and Ilya Brodsky, our CEO, also is contributing. So we will be using the T-Bioinfo platform to process big data sets. It's a comprehensive platform, cloud-based, and it's a very easy platform to use because students and researchers have been using it. So we have made it like in a way that it's easy to use. But it's a very powerful platform because it's integrating big data, and when we talk big data, we're talking about megabytes, gigabytes, terabytes of data sets. And it's really difficult to even visualize it. Like you cannot even open it in Excel. Just imagine how can you even thinking about analyzing the entire stuff. So you need a platform that can help you in that, or you can have it on your own computer if you have like a lot of hardware. You most of the things that are available are also available open source. So one of the purpose of learning is not to use this forever, but to learn the logic from here and understand that what all you can do and explore and while doing this. And if you find this convenient, you can use the TBioInfo platform. If you are into deeper research and want to start developing your own stuff, you're free to do that. So there is like a lot of 90% of the algorithm that we have used are open source. So everything is online and you can find them about it. So this is a platform, and this is the website. There are several sections in the platform. We will go to the platform after this, and we will start logging in. All of you will have an opportunity to go to the platform and log in, but there are several types of login. So we will have a demo login, which will give you access to the demo pipeline. So there are several sections that I was talking to you about. So NGS data is a type of data that can be analyzed using these sections. Mass spec is another kind of data that can be used that can be analyzed using these sections. And then structural biology data can be used we are still in development of a lot of these algorithms in the Tauber Bioinformatics Research Center in Israel, and in collaboration with multiple universities in the US. So one of those collaborations lead to this virology platform that is with UCSF. So a detailed virology section is there, which we will be using in infectious disease program. So that's a different program. And then there are data mining aspects, and you can learn about machine learning and application of machine learning using that. And then the individual user will have their own set of pipelines that they can see that what they have done, and their own login login access. Sorry. So if you are on laptop and if you are on desktop, then please go to this site and use this username and use this password to log in. And once you're logged in, let me know. So you can say 1 if you're in. And then we will start some further understanding, and do some hands-on stuff. Sorry. So I'm just keeping the screen on so that also Bhiksha has shared the password and the username in the chat. So you can just copy paste and try to log in. And once you're in, let me know. I also take a 1 minute break for questions. If you have any questions so far for what we have covered, you can ask that also. How much time do we have to complete the course? So the introduction to bioinformatics is a free course, and you can take your time. And then there are different also paid courses are there. So it's subscription based. So it's a 1 month subscription, and it's really there are like genomics, metagenomics, transcriptomics, and epigenomics. They are all available for like really 700 or 800 rupees a month, something like that. So we will share those by email. If you're interested, please you can check those and take as per. Please let me know if you're in or not. Nobody is in. Invalid. Type 1.
Shushmita, is it showing you invalid? Anyone else? It's same invalid. Okay, let us.
Okay, so we'll share another one. Try this one.
And when you are writing it in, please do not type it. Copy paste it and make sure there is no space before or after the username and password. Because if it shows invalid, often that is the issue. There is a space before or after the password, so make sure that it is not there. And I'll try logging in myself as well.
Okay. Yeah, but I mean, you can copy and save these in a notebook or somewhere, so that you can try that later, because at times what happens if like 40-50 people are trying at once,
Yeah, second
sometimes it
account
does
is
not work.
working. Yeah, everyone can log in via the second account, Dr. Mohit. It is working completely fine.
Perfect. Okay, so before I talk about before we go and do the hands-on practice, we will tell you about the data set. So it's important to know what we are doing and why we are doing it. So just an overview. So today we will use a high impact publication and the data that has been generated and submitted by the scientists and researchers associated with the article. So the article that we are going to look at is this one. It's a good example. This publication is interesting because the authors assembled a data set of about 50 different breast cancer cell lines that represents the entire, I mean, a whole spectrum of breast cancer variations. So, okay, this is the link to the publication. It's published in Genome Biology. It's been a while that it has been published. So I'm just sharing you the link of that article. And this is the article actually. So this is the publication, Modeling Precision Treatment of Breast Cancer. So the cell lines they have used have genomic data, transcriptomic, and proteomic data as well, and also an efficacy measurement of commonly used and experimental, drug, cancer drugs. So you can find it in this link that I've shared. If you're interested, please go through this publication. So here the data from cell lines and are combined with two types of phenotypes. One is clinical subtype, and other type is breast cancer cell line. So I'm not going to go into detail of this because we kind of covered this entire exercise in transcriptomics 3. But what I'm trying to tell you is that these authors have used machine learning. So these are machine learning methods, random forest LCM, on different type of cancer cell line data sets to find a way to have a personalized group or treatment for different patients, something like that. So even to think about that, I mean, how can you divide patients into different groups? So the first thing what a doctor does, I mean, when you go to a clinic, the first thing the doctor will ask you is he'll ask you about the information. He wants to know about, like, what is your age, and how much is your weight. Those are like phenotypic characteristics or clinical features. Those are also data points, right? Those are important for him to diagnose, because if it's a kid, if it's an adult, and if it's a woman, if it's a man, because the treatment can differ. And that's what I'm trying to tell you here. So the first important step is what? The first important step is diagnostics. I mean, diagnose. The doctor diagnose the disease. So once it is being diagnosed, then you are being prescribed a medicine, and if it is correctly diagnosed, you get well, right? So that is very important, to be able to correctly know what kind of breast cancer that person has. So what kind of, and when I'm saying what kind of, it means there are several types of breast cancer. There's not one type of breast cancer, there are several types of breast cancer. So they are called subtypes of breast cancer. And if I say these things, I think you will not understand. So let's look at it and let's learn from there. So let's take a look at real example. This is a breast and this is a tumor. And this is a mammogram, which is an X-ray picture of the breast that doctor used to look for early signs of breast cancer. Okay. So in this example, you can see the tumor is highlighted. Once it is found, it is critical to establish whether it is malignant or benign, and which tissue and what tissue it is originating from. Once the tumor is characterized by its phenotype, like size of the tissue and the origin, then we obtain additional information, like what kind of proteins are there. So to be able to do that, so you can see that different types. If it is inside, ductal carcinoma in situ, it's called when it's inside. When it's in a lobular form, so it's lobular. So there are different types. And when it is like in this ductal, but it's outside, so it's invasive. And same with invasive lobular, because these are obviously more dangerous, because it's going to infect a lot of other cells which are surrounding it. And then it is important to know what kind of breast cancer it is, right? So understanding what kind of proteins that are there to be able to diagnose or learn about the subtype. So over the period of time, this is a panel that has been developed, like this 20-30 years of research led to this kind of panel. So different type of proteins are there, like EGFR, HER2, Herceptin-2, BRCA1, and they are seen as a biomarker. So if they are present in that sample, and then we look for different types of expression of those proteins, then we understand that this person has what kind of breast cancer. So this is one way of diagnosing. This is called immunohistochemistry. So this provides us with only a selective number of biomarkers that we can check for what uh, if that is there or not. But oftentimes it's more than that, because there are new cancers you cannot detect through this, and if it is a specific kind, then only you can detect through this. Then we have microarray. So 50 genes panel. This is called PM50 panel. So this has housekeeping genes also, and this has those genes that causes that are biomarkers for breast cancer, different breast cancer. I mean, when you see a person having this breast cancer, you'll see high expression of that protein. So that's why we know that is a biomarker. So this has been established by studying different like studies that have been published from over the years, and people have come up with this panels to find out. So this is one way of doing it. And now the technology has evolved further, where we are looking at thousands of genes, because we are looking at the entire genome, entire proteome. So here you can see that they are differing from each other. There's a luminal A is one type, normal like is one type, luminal B is Herceptin enriched, basal. These are different subtypes of cancer. So in the data set that we are going to use, you will find these type of labels, because they are different type of breast cancer. And what we want to do, we want to see how they are different from each other, if they are separated, if there is a pattern or not. We are trying to find pattern, right? Because if we know a pattern, then we will be able to predict that if this is a breast cancer or not. But the challenge is, however, is to be able to deal with this data. So in the end, it looks like this. This is a gene expression table that we will be talking about in a while, and we will have the hands-on session just after this. But for you to understand and for you to be able to learn, it's important to know about this table. So this is the gene IDs, ensemble IDs, and this is the on the top you can see like different samples. So these are different patients. For example, these are different patients, and these are different genes and their expression. So one patient will have a different expression, one patient will have a different expression, and like this there are 20,000 rows. So this insane number of rows or columns. So that's the not 20,000, actually it's 80,000 or something, or 100 million, 100,000 number of samples. So there's like tens of thousands of samples and complex relationships. How would you learn that how a group is, what is the pattern of this group? So this is a challenge, and this is what we are going to address in this session, that how can we understand and interpret such kind of data to be able to find biomarkers, discover new biomarkers, and we will be using machine learning. So one of the example is PCA, principal component analysis, from table of expression to patterns. And to take you through this entire experience and do the hands-on analysis, I would like to invite Dr. Harpreet Kaur to take you through this practical experience. Over to you, Dr. Harpreet.
Thank you, Dr. Mohit. Let me share my screen. So, welcome everyone. So, let me give you a brief introduction about me first. So, my name is Harpreet Kaur, and I have a PhD in bioinformatics, and my research is on biomarker discovery for diagnosis and the prognosis of the liver cancer. So in my current role, I am Omicslogic Bioinformatics trainer with Biomedical, and I'm happy to assist all the participants to learn about data analysis, machine learning, and visualization in the application of large data sets used in the biomedical research. So let me take you to the T-Bioinfo server and will show you how we can produce a well-structured gene expression data table that already shown to you by Dr. Mohit, and how we can produce that kind of table from the raw sequencing read data in simplest way using T-Bioinfo platform. So let's go over the platform. So if every one of you able to log into this server, just let me know.
Dr.
So if you are on this,
okay.
So if you are on this server, so here you will find the different options on our platform. So if you look at here, we have a NGS data, have a section on the NGS data analysis, then mass spec data, structural biology, data integration. So here, as Dr. Mohit have shown you that how we can produce a gene expression table when we have a raw sequencing read. So first of all, why we need a raw to process that data? So even a single sequencing read. So those sequencing reads are present in a FASTQ file. One single FASTQ file is it's like in the GB. It could be 8 GB. It could be a 10 GB. So even for a single sample. So eventually, how we can process that data? So under the NGS data, here you will get an option that is transcriptomics. So under the transcriptomics, sorry. So under the transcriptomics, here, so, just give me a second. Just give me a second. I just logged out from this server. So just meanwhile, just let me know how many of you able to log into this server.
The server is down, by the way, Dr. Mohit. So maybe we cannot have a hands-on session today.
Oh, that's a very unfortunate incident. I think it's just.
Yeah.
Okay, let me check as well.
Down. Yeah, it just suddenly went down.
Okay, it's just okay. That's fine. Actually, we can do the hands-on on the code platform if you can try.
Okay, okay, yes. I guess that's working fine.
Yeah, code will be working fine.
Okay. So as Dr. Mohit have told you, so okay, let me share my screen again. So once you have that table, so that table will be obtained by running a pipeline on T-Bioinfo server. So once we have that table, so then how you can perform any kind of analysis. So this is our another platform, that is Code Omicslogic platform. So on the Code Omicslogic platform, here we have various courses which will explain you what were the logics that are when we are running the pipeline. So whatever the logics and how all those logics works if you look at into the code. So at here you will find the different courses for different kind of analysis. So just let me go to the introduction to the data science. This is one of the course which will explain you the different lessons, like how you can load your file, how you can visualize your data, then how you can apply machine learning, or how you can apply statistical analysis. So under each and every lesson, here you will find the different lessons. So let's go to this dimensionality reduction and the PCA visualization. So whenever you are in on any lesson, so the first section always in each lesson, here you will have the first lesson, the first step would be the understand the problem. So first of all, it will explain why we are going to run particular kind of analysis. For example, this is a dimensionality reduction using PCA. PCA is a principal component analysis, which is one of unsupervised machine learning method. So the first part that will give you the explanation what kind of data that we are going to use and what kind of analysis that we are going to run under this particular section, and what were the outcome that you will be expecting. So all those the logics will be explained in the first section. Then the second part, that is learn the code. So here you will find the different code blocks that will explain each and every step that are we are going to use in the in our code. So it will explain how you can load your particular library, then how you can load your data, then how you can generate a matrix, then how you can transpose a data for particular kind of analysis. Then if you are running a PCA, that is principal component analysis, so what would be the function that we are going to use. So all, and finally how you can plot your PCA plot. So all these explanation is step by step is explained in this subsection, that is code breakdown. So once you learn about coding, that how it works, then the third section, then in the next section that we have, that is understand the analysis. So once you done that, then you just complete and go to the next. So in the next part you will get all complete code. So now let's see how we can run this analysis. So in the first step, if you look at this, so always the first step always provide you the explanation exactly what are we are going to do. So like here, first of all we are giving input file, then we are defining how your data should be defined, then we transpose that matrix. So once you run this, then we are with PRC component, that is a one of the function that is we are going to use for the PCA. So then with the it will produce your PCA components. Once you have those PCA components, that will be stored in the object that is called PCA. Then how you can plot it. Now let's look run it and see what we will obtain. So once you run this code, and you will get the results that here, if you look at this. So here we will able to obtain a graph that will explain the principal component analysis. If you look at this particular graph, so it will show you this is the plot between the principal component 1, which explain the nearly 50%, 55% variance in the data, and between the second component, that is PC2. So that is principal component 2, that is explaining nearly 19% variance in the data. So eventually you're able to obtain this kind of plot, which will explain how your samples are actually getting modified. So beside, so if you want to suppose here you have all you can modify this code. So this is not just the lesson. So here you can modify your plot. So then suppose I am just want to add to color. I want to add. Let me just demonstrate you here. So first of all, here we are going to load our data. So first of all, we have requires two libraries. One is G45, second is cluster. So let's run it, and then we will load our data. So once you run it, you will get a pop-up success running. So then let's load this data. So the input data. So in the R, we read our data with the read.table function, and then we will just let me. So here I am posting the link of my file. So then we have to define what is the separator. So here separator means how the columns in the file are separated from each other. So they are like separated with the tab. Then we have to define whether we have the header in the file. So if there is header in the file, that is whether the header is present. So if you look at this file, that is here I'm providing in the first row, here we have the IDs, and then we have the followed by the gene expression label. In the columns we have the samples. So then even we are also defining what kind of sample it is with the group. In the second column we are defining that. So then in the first row we have the sample IDs. So here we have to define that header equal to true. Then it is also asking whether row dot names equal to 1, that whether our data have rows. So with the as we have already shown you that in the first column on we have sample names, so we are defining row names equal to 1. Then we are also defining check dot names equal to true, false. So this is how we load. Now once we able to load the data, now how we can check whether our data get uploaded or not. So with the simple command, that is dim dimension, this the dim the data. So now you can check whether your data get loaded or not. So it will give you the dimensions, that how many rows and how many columns you have in your data. So first it will indicating there are 52 rows are there and 16 columns are there. So then how we can run the, then as our the second, if you look at this data here in the second column we have sample names. So whenever we plotting any graph in R, so it will not take any, it will take only integer values. So these are non-integer values, so we have to remove those non-integer values. So what I will do, the data, I am making a new data frame, new object. So where I am just taking my data from the second column. So I'm skipping that first column. So I have total 16 columns. So I am just transferring my data to a new data frame where I have removed the column which contains sample names. So once I'm done that, now I can check even the dimensions to check, to make sure whether the column is removed or not. So how I can check the dimension, that is data 1. So if you look at the this dimensions here, what you will get, that here we have the 52 rows and the 15 columns. So our second column get removed. So beside you can also check it by doing the head. The head command will do what it will going to do, that it will show you top 10 rows. So if you can check that as well. So if you look at this here, you will get able to print this, and you know that you now you have only ensemble IDs and the sample IDs. So once you're able to do that, then we are going to make a PRC components. So PCA component, here we are going to make a, we are going to run PCA component. So with the PRC comp data 1. So here what we are going to, we are doing that our on we have able to obtain a data where we have only integer values, that is we have the gene gene values, gene expression values corresponding to the samples. So we with the PRC comp command we are going to generate PRC components. So I am storing this PRC component into an object that is called a PCA. So then I can plot this PCA plot. Sorry, PC1. I have to. I just. So if you look at this, with simple, first of all, we just generate with PRC-COM, we have able to generate the PCA components. Then with the auto plot, with the PCA1, the object where I have stored my PCA component, with the auto plot, I am able to produce a PCA component. So now if I want to color, I want to check these data points, what are they? So these data points are actually representing the sample. Now if we want to check which category it belongs to, or which class or which cancer subtype it belongs to, so how we can do that? If you look at this data here, you will get that the class of the sample is actually given by the group. So if we able to so here we have to define, oh sorry, so if I do the auto plot, then I have a PCA 1 color equal to so rainbow length, and I have to define the group. So where I have the group in the data dollar group. So meanwhile, I can define the color from here as well, direct. So just give me a second. Okay, just okay, sorry. Just here I have just added two brackets. So with the color, it should not work. Yeah. So I just added by mistake just two brackets. So we have to make sure that there is not bracket. So with this, we can give the color. So with simple color, with the color command, so then you can also decide the if you want to use this color with the other command that you want to use the group. So this is a simple way where you can learn how these codes work. So you just have to take care, like here I just by mistake I added two brackets, so this code was not working. So you have to take care of all those things. Beside, we can also decide the color based on the groups. So these kind of exercise that you can do on the this code OmicsLogic platform. So you can visualize your samples. So this is the simple best way.
Yeah,
Yes.
thank you, Dr. Harpreet. So we do have a few questions in the chat box. If we can address those and move forward.
Okay.
Yeah, so the purpose of this entire exercise was to show you that it can be done through a intuitive platform. And to answer Prithviraj, do we have to do the same coding? So coding is absolutely optional. So the T-Bioinform platform gives you the output, and then you can play with that output. You can do several things with the output, and all of these tutorials and everything is described in all the courses. So whatever we did, it's described in transcriptomics 1, 2, and 3. So we covered a lot today, because one of the reasons that you have not been able to understand all of it is the reason of is that reason. So what is required after this is you to go to these resources and try it yourself. Like once you try it yourself, you'll find that many of the things are explained in text. So you don't have to you know uh, learn it right now. You can learn it at your own pace, because of these are made into courses. So everything is made into courses and explained really well. And then we are trying to demonstrate you how do you go about it. And the TBioInfo platform does not require you to do any coding. It's built for biologists. But if you are interested in coding, if you want to go beyond doing the analysis and do some downstream analysis, then you can try coding. And again, it's an option that you can avail, because what we are doing is that educational platform. You're learning everything, you're looking at examples, you're doing practice hands-on on examples and getting all that understanding. And then you are doing exercises on TBioInfo platform, big data sets you're analyzing, you're getting tables, and you're looking at those tables and analyzing those tables using machine learning algorithms. And everything is point and click. So this is just what you can see, as you saw in the TBioInfo platform, that there are pipelines, workflows that are explained very well, and you can totally do that analysis by your own. And everything that we do has a component of mentor support. So online, you can write it in the forum, you can email us, you can put it in WhatsApp. We have group in WhatsApp, we have a group in Telegram. So there's a huge platforms to have a communication, to overcome all the problems, all the questions, all the confusion. So that the purpose of this entire thing is to be able to guide you and train you, and to be able to make you efficient in bioinformatics, that you don't fear bioinformatics. You see data, you understand that, okay, this is the analysis they have done. This is what they might have come up with. So you have a understanding already. That kind of thing is a development that is really very important in education. And learning about publications and research topics is really exciting, I guess, if you're interested in biology and if you're studying this, and if you see that yourself doing something about it and having a future in biology or medicine or anything. So I think it's great to know all the technologies and the data, what kind of data that is being generated, and the stuff. So thank you, Dr. Harpreet, for showing this. And as I was telling you that that you have to practice and look for an email from us on Monday at marketing@find.bio. So we will email you the details about what we have covered, this session, recording of this session, and a video of the RNA-seq pipeline that we could not do today. So we will send you that video that is already recorded. So there's a question from Prithviraj Paul. One question is to me, is if time permits, is that you are a data scientist. So how did you approach it? Meaning, which courses have to be taken after MSc to become a data scientist? So I think you can start right now. I mean, to be able to do research is to do PhD. So I did PhD to do research, but at that time I have no idea about this platform and this technology being accessible to students. So it was accessible to researchers who has access to a research lab, a workstation. So those are the people who were able to do data-driven research. Now with this technology, cloud-based technology, everything is available to everyone. So sitting at home, you can actually do this research. You can start right away to become a data scientist. You can take these courses to start off with, and then there are so many content and projects. So important thing is just to not learn this, but to apply this. Once you start applying, you become a data scientist. So application is in project. What we do in our online programs is that we teach this like weekly, bi-weekly sessions. Every week we have two days dedicated, 1.5 hours each, to learn this, and then there are these courses that you have to complete alongside. And then once your learning is complete, you propose a project. Either you propose your own project, or you propose a group project, or a university project, whatever bioinformatics project, uh, based on the thousands and thousands of data sets which are out there. So in the program, we do that as well. So to tell you about the upcoming opportunities, so if I can share my screen, just to tell you about some of the upcoming opportunities. So we have a program starting in Kolkata, especially for people who are in West Bengal. So this is designed for students in IG, Institute of Genetic Engineering, Madhyamgram. But this will be an entirely online program and will be available for other students to join also. This will be a 6-week program. It will have only 6 sessions. Every week there will be one session on Tuesday, 9 a.m. So just one every week, and then there will be these courses, and all the access will be there. Your personal access will to T-Bioinfo platform, your personal access to the code platform, and it's really an affordable course because it is designed for students in Kolkata. And we will send you the details, and the date is still kind of tentative. It's not 100% finalized, so that's why I'm not sharing more details. But we'll send you the via email. So look for email. So this is a course which is going to be taken by Dr. Harpreet. I'll be there, and Dr. Kamalesh will be there. He's a faculty in IT Kolkata. And another program, if you're interested in virology, if you're interested in doing something in COVID-19, if you really want to do do a research on COVID-19, so this is a great program to join. This is an international program, and it's an Omicslogic international program that will be conducted online, entirely online. And again, you will have access to selected courses that are relevant, selected projects, and selected I mean, you will have an educational access to the T-Bioinfo platform also. And this is also being provided to Indians at a sustainable registration fee. So what we are trying to do here is to bring it to more and more students and make it as sustainable as possible, because we have to teach, we have to maintain the cloud resources. So just to able to do that, there's a fee, or else it would be free. But it cannot be free because there are efforts of people doing this in and out every day, and that's impossible to be sustained without having a fee. So there's a small fee. This is again a 1-month course. The other course is 6-week course, and there will be another 2-month course which will be about just the materials and few sessions. But this course will have 10 sessions, and this is quite intense, and you will be able to work on infectious diseases in depth. So we will be sending you all these details, and for today I think it's time, and it's the 90-minute session, and we are reaching there. And if you have any more questions, please go ahead and ask. And I would again like to thank you all for this, for your time, and for you that you did all here trying to learn with us. Really appreciate that. And if you have any questions, you can reach out to us at marketing@pine.bio, and then there are several other options as well. So there are LinkedIn, where we keep on posting jobs related to this industry. And for people like students like Prithviraj, I think please join the LinkedIn group and Facebook group, because you will get to know what kind of jobs are out there and what is happening in this field, because we try to publish articles there. So if there are no more questions, I would so yeah. Please explain what is PC principal component and how do we analyze it? Okay. Okay, Prithviraj. So we have a minute. I will just take a minute or two to do this because we are running out of time. And principal component analysis is a machine learning method, unsupervised machine learning method, and it's really difficult to explain this into in one session, like we tried to do along with other things. Dr. Harpreet also tried, but actually it takes a little while to learn about this. So this is a slide deck that I used to teach machine learning. So PCA is there in the T-Bioinfo platform. This is the interface. As I was telling you, it's point and click. You click here on start, then you choose this and then end. Then that will do the PCA. But there are some things that you need to learn about PCA. So what PCA is doing is that if you see this box, there are so many data points in there, and you can see this in a three dimension. So PCA is doing what? PCA is trying to make some meaning out of your big data. So your data is so big that it's impossible to visualize and understand the meaning out of it. So what we are trying to do is dimensionality reduction. We are reducing data to the important part of the data. What is important part of the data? First of all, we want to see the variation. That's why Dr. Harpreet was saying variance. Variance means variation. Matlab, what are the differences? What are the differences between two people? What are the differences between two patients or a group of patients? So we want to see how different they are. So we are in PCA. What is happening in normal things in that if you have two genes, you can plot it into a two-dimensional plot, and you can see how they are lying, right? If you have three genes, you can plot it in a three-dimensional graph and see how it is looking, and if there are patterns or there is a line that you can draw and separate them or not. But if you have like thousands of genes, then how do you plot? Beyond three, it's not capable for humans to see this, right? So if you have multi-dimension, a problem of multi-dimensionality, so this is to solve the problem of multi-dimensionality. What it is doing, PCA, let me tell you. This is when the data points are close, right? So PCA does what? PCA takes all your dimensions from the data and change it into this kind of a plot. So what it is doing is that it is calculating variance. Okay, matlab the differences. If the data points are together like this, it is clearly low variation. Na? If there is if it is all around, there's a lot of variation, you can say. And if this is like this, it's low variance. If the data is like this, this is high variation. So what is PC1? PC1 is from this line to this line. What does this mean? That this point to this point, there will be the most variation. Is it clear or not? This is pretty simple. If I'm not explaining it right, that's another issue. But from this point to this point, there's a lot of variation, right? This line will capture the most amount of variation, right? So that's why it's PC1. As Dr. Harpreet was saying, PC1 captures the most variation, and then PC2, and that likewise there will be PC3, 4, 5, 6, 7, 8. So the variation is captured like this. So the most variation in your data you will see in PC1 plot, and PC2 as henceforth. And then when you rotate it like this, it comes up like this. This is your final PC plot that we were seeing. So this is how we can run the PCA. And this is the kind of result that you get. This like different colored. We did coloring and stuff using the code platform that you saw. That you can change the color of your samples and see how they are lying in your graph. So this is a typical graph, and what you can see in this graph. I mean, let's interpret. You saw there are 4, 5 type of breast cancers, right? You saw basal was there, Claudin low was there, luminal was there, normal like was there. So what it is showing, there are colors. So luminal, you can see luminal are all separated into one area. If you can draw a line, you can divide luminal and others. So it means that luminal breast cancer, luminal wala subtype, has a pattern that you learn by doing PCA. Is it clear now? So you can obviously do this kind of stuff on the code platform and see how they are grouped. You can
if I
what
may
we
add,
did.
Dr. Mohit, yeah, so these all would be covered in details in the coming sessions. So if you have any questions, you can join us there, and we will be covering all of these topics in details during the upcoming programs, both in data science for biologists as well as bioinformatics for infectious diseases. So there you can join these programs and you can go through not just learning about PCA, but also start from very basic courses that will help you to learn more and go towards PCA and more advanced topics. So please come and join the sessions if you are interested in learning about these topics. Yeah, thank you so much, Prithviraj, and thank you, Dr. Mohit and Dr. Harpreet. It was great to have everyone here. If you have any more questions, please email marketing@pine.bio for any questions that you may have.
Yeah, Deepsa,
Yeah, over time, Dr. Harpreet. Yeah.
okay, can you give me for 1 minute? So let me just show you the already Dr. Mohit has